With the advent of mass production, our society has come to depend on a continuous and reliable supply of identical consumer goods, one item after the next, an identical copy. It's a recent feat of technology, but it's one of the oldest concepts in biology. From the very beginning of life, individual cells have replicated and reproduced themselves, creating exact duplicates. Organisms grow cell after cell from a DNA blueprint in the nucleus that is copied exactly and passed on to each new cell. This process of nuclear division is called mitosis, and the division of the cytoplasm is called cytokinesis. Cell division is an important part of our biological heritage. In this program, you'll examine the life cycle of the cell, which includes the process of cell division. You'll observe the behavior of chromosomes during mitosis and the division of the nuclear contents and cytoplasm to produce two identical cells. By the end of the program, you'll be able to explain the events of the cell cycle and how they apply to growth and repair of aging or damaged organisms. And you'll see how science and technology are being used in the fight against cancer. What are you and Cam looking at, Bibi?
It's epithelial tissue magnified 400 times. Pretty good view.
Are there any cell structures on the slide that either of you can identify using this model?
Well, there's the nucleus, there, the nucleolus, there, and the cell membrane, just right around there. Now, there's also some dots in the cytoplasm, but that's about all you can see.
That's pretty good.
I have a plant slide. It's a cross-section of a leaf.
Well, why don't you put it on the light microscope and we'll have a look.
Sure. Okay, I've got it.
Oh, that's it.
I can see some of the same structures as Cameron.
Can you point them out on this model of a plant cell?
Okay. There's the nucleus, the cytoplasm, the cell membrane, the cell wall, and all these round structures. The green ones here in the model.
Chloroplasts.
Thanks. I can see them almost as easily as the nucleus.
That's pretty good, Bibi, for a first glance. We need to look more closely.
How close?
Well, let's try magnification of 25,000 times. These are photomicrographs. They were taken using an electron microscope. A nucleus is found in almost all cells. It is often called the control center of the cell because it contains the genetic information that directs most of the cell's activities. This genetic material is in the form of long, thin fibers of DNA called chromatin. Also in the nucleus are one or more nucleoli. They appear as dark spherical bodies, and they function in the assembly of ribosomes. In animal cells, but not plants, centrioles can be seen. They appear as a pair of objects close to the nucleus and are made up of microtubules. The centrioles are involved in mitosis. That's what the nucleus and the surrounding cytoplasm of a cell looks like. What's its life cycle like?
Well, the parent cell divides, and then the new cells either may or may not divide themselves.
But before they divide, what's happening?
Well, they have to get food.
And oxygen for cellular respiration.
And they must grow.
And make protein and other substances.
But they can't get too big, though.
Right. A cell does need a large enough surface area relative to its volume to allow the efficient transport of materials in and out of the cell. The life cycle of a cell is the time elapsed from one cell division to the next. In actively growing mammalian cells, a cell cycle lasts from 16 to 24 hours, although there may be wide variation from less than 8 hours to more than a year. Mitosis and cytoplasmic division together take only about 1 to 2 hours. During the remaining time, the cell is in interphase. Within interphase, there are 3 stages of growth: the G1 or gap1 phase, the synthesis or S phase, and the G2 or gap2 phase. In G1, the new daughter cells increase in size and produce more organelles. They manufacture enzymes and other proteins for metabolism and other cellular functions. If these cells go through another cell division, they also manufacture materials needed for DNA replication. In the S phase, DNA is replicated for the next mitotic division. In the G2 phase, organelles and enzymes are manufactured for the upcoming mitosis and cytokinesis. Accuracy of DNA replication is checked during G2. The cell repairs any mistakes in DNA replication before beginning to divide. By the G2 phase, the cell has grown considerably in volume. Cell division restores the larger surface area to volume ratio. The duration of cell cycles varies considerably depending on the cells. The cell cycle of bone marrow cells is about 10 hours. Most adult cells, such as nerve and muscle cells, are in a special resting phase of the cell cycle called G0. In G0, cells are carrying out their specialized functions, but are not preparing to divide again.
I understand that you're studying cell cycle and cell division. We're going to do an experiment today to actually explain or have you see how cell division actually occurs in plants. We've actually chosen to use the plant root tip. And if we look at this model of a root tip, you see that it has a cylindrical shape. And the idea of the preparations that we will do here is to dissolve the cell walls so that we can smear out or squash the cells into a single layer. Now here we have lettuce seeds that we've germinated for 48 hours. We take 2 or 3 of these lettuce seedlings, put them into a watch glass, and then add equal portions of 1 molar hydrochloric acid and a dye called acetoorcine. The hydrochloric acid dissolves parts of the cell wall so that the cells can separate nicely when you do the squash, and the acetoorcine is the dye that we use to stain the DNA. We have to put the watch glass on this warming table at 60 degrees Celsius for about 10 minutes. The hydrochloric acid and the acetoorcine at 60 degrees will speed up the process of separating cells and staining the DNA. The 10 minutes are up now, so let's complete the preparation of our slide. I'm going to remove just one of these seedlings that we prepared earlier and cut off just the bottom 2 millimeters of the root tip and discard the rest of the seedling. Then I'll add 1 drop of fresh stain and use a dissecting needle to squash that root tip in that stain. Then I'm going to apply my cover slip, fold over the paper towel, and then place my thumbs on top of the cover slip and squash the cells of the root tip. Cam and Bibi, why don't you try one of your own slides?
In some of the cells, I can't see a nucleus. Instead, I see these little tiny threads. What are those?
Those are actually the chromosomes composed of DNA and protein.
I could see something like a line in the middle of the cell.
I see that too. Do you think that's a cell wall?
What would a cell wall be doing in the middle of a cell?
Well, actually, we call that a cell plate. And that's how plant cells separate the two daughter nuclei after mitosis. And that's a process called cytokinesis. If we look at the monitor of one of the slides that we prepared earlier, these two cells here have divided, and you can see a faint line running between the two daughter nuclei. That is actually the cell plate, and that means that cytokinesis has occurred separating these two daughter nuclei.
Why can we see the chromosomes in the slides we looked at in the first lab?
Those particular cells were in a stage called interphase. And when cells are in interphase, the chromatin is stretched out into a long, thin thread, and it's all balled up together, very much like a ball of yarn. And that means you cannot see individual chromosomes. But as we begin to go into mitosis, the chromatin condenses into the separate and very recognizable chromosomes.
I noticed something else, too.
What's that?
In some of the cells, the chromosomes were bunched together near the middle of the cell, and in others, they were closer to the outside of the cell. It was like they were being pulled apart.
That's correct. That's a very good observation. Again, if we look at the monitor, we can see some of the cells that you're probably seeing in your own preparation. This cell here shows the plant cell in metaphase, where the chromosomes are at the middle of the cell. And here, this cell shows them in anaphase, where the different parts of the chromosome, the chromatids, have been separated from each other.
Mitosis is a series of events that causes a cell's chromosomes to be divided. The process of mitosis takes place in 4 phases: prophase, metaphase, anaphase, and telophase. Specific events occur in each phase. There are no pauses between them. The process is continuous. Mitosis is usually followed by cytokinesis, the division of the cell's cytoplasm and newly formed nuclei. When the chromosomes become visible at prophase, they have already been duplicated. The new chromosomes are actually made up of 2 chromatids. Each chromatid pair has identical sets of genes. The 2 identical chromatids are attached to each other at a point called the centromere. If you want to count chromosomes, a good rule of thumb is to count centromeres. If there is only one centromere, there is only one chromosome. In prophase, the centrioles in animal cells are also seen moving to the poles of the cell, forming the spindles. The nuclear envelope breaks up and the nucleoli disappear. In metaphase, the asters are pushed further apart by the mitotic spindle growth. The chromatid pairs move to the center of the cell and can be seen attached to the spindle fibers. Spindle fibers attach to the centromeres and are responsible for separating the chromatids during mitosis. Other spindle fibers reach from pole to pole or radiate outward from the centrioles. Anaphase begins with the centromere splitting, and each set of daughter chromatids, now called chromosomes, are pulled apart to opposite poles by the shortening of spindle fibers. In animal cells only, the cleavage furrow forms. In plants, the cell plate can be seen. By telophase, the daughter chromosomes are completely pulled apart and are at the poles of the cell. The spindles disperse. A nuclear envelope forms around each set of chromosomes. The chromosomes become more thread-like as they uncoil, and the nucleolus reforms. During anaphase, cytokinesis begins. For plant cells, cytokinesis involves the formation of a cell plate in the center of the cell. Carbohydrate molecules fuse together to form a new cell wall. Cytokinesis in animal cells is different. Microfilaments, which form a ring around the equator of the cell, contract and cause a cleavage furrow to form. The cleavage furrow deepens and pinches the plasma membrane in half so that 2 new cells are formed.
As scientists often use models to help them understand a concept. I've brought in this sheet of Bristol board to represent the inside of a cell.
Okay, we can do that. We can use these pieces of yarn to represent chromosomes. And that would mean this is a cell with 2 chromosomes.
Okay. Now in S phase of interphase, these chromosomes would have doubled. And I'll represent that by placing an identical piece of yarn next to the original.
But this isn't visible until prophase.
Right.
The centrioles become visible during prophase as well. That's where the spindle fibers are formed. We can just imagine the spindle fibers. It'd be too difficult to draw them. But the spindle fibers will attach to the centromeres of these chromosome pairs. I'll replace these yarn chromosomes and put these marshmallow centromeres on.
Okay, now it would be metaphase. So the chromosome pairs would line up on the equator of the cell. Like that.
And here comes anaphase. I'll need some help here. So if each one of you can grab a centromere. Okay, the centromeres divide and the chromatids are pulled to opposite poles. The result of this is there is the same number and type of chromosomes at each pole.
There's also the same number and type of chromosomes as we had in the original cell.
And now the chromosomes would revert back to their original form of chromatin.
But what about a nuclear envelope?
Well, 2 new nuclear envelopes would form, one for each. And then cytokinesis would complete, and 2 new cells would be formed.
Houston, we have mitosis.
Our understanding of cell reproduction raises the question, why do cells age? Studies have been conducted on astronauts in weightless environments to measure the rate of aging and deterioration of cells. The evidence indicates that inactivity promotes more rapid cell deterioration. This is supported by hospital studies, which suggest that a reasonable level of physical activity helps patients recover more quickly from illness or injury.
In my opinion, and in a growing number of people's opinion, aging can almost be looked at as the flip side of cancer. An aging cell has a natural limitation to its growth. It will divide a number of times before it stops and becomes old-aged or senescent. A cancer cell grows along, and somehow it breaks through those boundaries and becomes immortal. If we understand the genetic mechanisms that limit the growth of normal cells as they become senescent, we'll obviously understand those limitations that are gotten around by the cancer cell, mutated somehow in the cell, and contribute to the formation of an immortalized cancer cell. We're doing research primarily using the so-called fibroblast cell type model. These cells grow in culture, they go through a certain number of population doublings, and what we're doing is trying to dissect the genetic program that is responsible for blocking their growth. We have models that are normal cells. In fact, I've given cells to the cause, and some experiments are done on my cells, and we've picked on some people on the floor as well. And we also have very defined cell lines from the American type tissue culture collection in the lab. What we're able to do with these cells is follow them as they go through their in vitro lifespan, as they use up their lifespan. And we also have cells that are basically the same type of cells, but they've been immortalized experimentally. We can take certain viral proteins, put them into these cells, and the cells become immortalized. When we look at the immortal cells and the normal cells that are capable of growing and the senescent cells, all of which are very, very similar, we look for changes in gene expression that might be responsible for the immortalization of the cells and the natural limitation of their lifespan in the senescent cells. Another set of experiments that we're doing presently is to try and figure out how the ends of the chromosomes, the telomeres of the chromosomes, may contribute to limiting the lifespan of cells. And that's a very new area, and it's causing an awful lot of excitement. In fact, there are a couple of biotechnology companies that have grown up around these very same ideas in the last couple of years. We're trying to look at the blood cells. This is another natural aging cell type in the body that goes through a certain number of population doublings before it stops. Good evidence of the blood cell model is what's seen with the immune system.
Certain factors, such as chemicals or hormones which stimulate or inhibit cell division, have been identified. Although scientists do not fully understand how these mechanisms work, signals in the G1 phase seem to play a crucial role in determining whether a cell goes through division or switches off. In the case of certain cancers, cells tend to divide repeatedly, which disrupts and can eventually kill the organism. Continued research on the mechanisms of cell division could eventually be applied to cancer prevention.
Interesting. What are you working on now?
We heard you could figure out how long it takes cells to complete mitosis.
We looked at 50 cells and we counted the number in each phase. Here we have 33 prophase, 12 metaphase, 2 anaphase, and 3 telophase.
And if we assume that it takes 60 minutes for onion root tip cells to complete mitosis, and that the number of cells we counted in each phase is a measure of the time required for that phase, then 33 out of 50 times 100, which is 66% or 40 minutes, is spent for prophase, 24% or 14 minutes in metaphase, 4% or 2 minutes in anaphase, 6% or 4 minutes in telophase.
Good. To be accurate, you should do several more trials and average your results. And remember that the duration of phases will vary depending on the organism and tissue. How would you calculate the time taken for interphase?
Well, if we took 5 fields of view, we could then count the number of cells in interphase and the number of cells in mitosis.
And if we had 4 cells in mitosis and 46 in interphase, we would do the same sort of calculation. You would take the fraction of cells and multiply it by the total cell cycle time. If the total cell cycle time is 10 hours, then 46 divided by 50 times 10, or 9.2 hours for interphase, 4 divided by 50 times 10, or 0.8 hours for mitosis.
Yes, that would work well. Tissue repair or wound healing occurs by regeneration or by fibrosis. In regeneration, destroyed tissue is replaced by the same kind of cells, whereas in fibrosis, repair occurs by the formation of scar tissue. Which of these occurs depends on the type of tissue damaged and how severe the damage is. Generally speaking, clean cuts heal more successfully than ragged tears.
What happens if cells don't stop dividing, or if they're dividing faster than the other cells around them?
A tumor or cancer is formed.
But what causes it?
That's the big question. It's still the subject of considerable research. We know that it involves changes in genes, such as the abnormal activation of the genes that control growth and mitosis.
So in cancer, what happens is you get excessive production of growth factors causing excessive proliferation, or you get loss of inhibitors which allow mitosis to continue. And in the really malignant ones, you generally have both factors. You have both stimulating factors to growth and you have loss of inhibitory factors. So those are the major things that make a cancer cell differ from a normal cell. What we are looking for as a pathologist when we examine it under the microscope is how it differs from the normal tissue, okay? And one of the easiest ways to determine how it differs is by examining it for mitotic figures. In a normal tissue that's turning over at the normal rates, you rarely see mitotic figures. If you've got a cancer, you get increased numbers of mitotic figures, and the more rapidly it's growing, the more mitotic figures you get. The commonest treatment is still surgery to remove the tumor. But in many instances, that's totally inadequate. So nowadays, we generally supplement them with 2 major kinds of treatment, radiation therapy and chemotherapy. Radiation is exposing the tumor area to X-rays, of a certain kind of X-rays. The effect of that is not to kill the cell immediately. It releases free ions, free radicals within the cell, which attaches to the DNA, damages it, so that the next time the cell is ready to multiply, it can't do that because the DNA is ineffective now. So in that way, it can stop a tumor multiplying. And the more rapidly a tumor is growing, therefore the greater the effect of radiation will be in those instances. Now, what you have to be careful of with radiation is that any normal tissues, which are turning over rapidly will also be affected by the radiation if your field is not very confined. And that's why you get the side effects of radiation. The lining of the stomach and the rest of the GI tract turns over very rapidly, renews itself very rapidly. So you often get nausea and vomiting associated with radiation treatment. Similarly, your bone marrow is turning over very rapidly all the time. After radiation, they often get a low white count, which makes them more prone to infections. Chemotherapy, there are a large number of drugs available nowadays that affect different kinds of tumour. The vincristine attaches itself to microtubules which make up the spindle that forms during mitosis. So when that happens, of course, mitosis can't occur in the normal fashion. It becomes very abnormal. You get very bizarre shaped mitotic figures when you see it under the microscope. And of course, the resultant cells are even more abnormal and they can no longer proliferate. So in that way, you can stop the growth of tumour. Another example of a chemotherapeutic agent is methotrexate. It's used for a lot of tumours. It's used for some leukemias as well. It's used for a lot of brain tumours. It's used for testicular tumours and many varieties. It acts differently from vincristine. It's called an anti-metabolite. The reason being is that the drug attaches itself to DNA and prevents the DNA from forming, multiplying, so that you can't get replication of your chromosomes. Okay, so you no longer get mitosis, so the tumors stop growing. So those are good examples of how treatment can affect the mitosis of normal or abnormal cellular proliferation.
In this program, we've seen how two identical cells are formed by the orderly processes of mitosis and cytokinesis. We've looked at the life history of a typical cell and seen how the events of the cell cycle are important for mitosis and for the functioning of the cell following its division. In our own bodies, the trillions of cells arose from a single-celled zygote and then differentiated into various tissues with specific functions. Now that's a lot of mitosis. And that process continues until death. We are continually producing new cells and replacing old ones. As science furthers our understanding of cellular reproduction, there are increasing medical and technological applications. From the mysteries of cancer to the reality of aging, the cycle of cells is a key to the cycle of life.